My name is Dr. Celeste Stewart. I'm a patient engagement manager for the National Ataxia Foundation, and it's my privilege to welcome you to the session all about SCA10, or more specifically, research on SCA10. It is my privilege to introduce our speaker today, Dr. Mario Cornejo-Olivas. Dr. Cornejo-Olivas is an attending neurogeneticist and senior researcher at the Neurogenetics Research Center in the Instituto Nacional de Ciencias Neurológicas and a research professor at the Universidad Científica del Sur, where he leads the neurogenetics working group in Lima, Peru. He currently serves as the president of the Counseling Committee for Rare Diseases of the Peruvian Ministry of Health. And Mario runs a neurogenetics outpatient clinic primarily focused on inherited ataxias and other repeat expansion disorders, which was recently recognized by the National Ataxia Foundation as an Ataxia Center of Excellence. His research on SCA10 and other inherited ataxias has significantly advanced the understanding of the genetic epidemiology of Spinocerebellar Ataxias and other inherited disorders in Latin America. He also oversees the Peruvian DNA Bank for neurogenetics research. Mario is honored to serve in various roles related to his work on ataxias and other neurogenetic disorders, including as a faculty member for the national and international scientific events. He has published over 70 peer-reviewed papers in high-impact journals. And with that, the floor is yours.
Thank you, Celeste. It is a great privilege and a great honor to be here and to have this opportunity to share with you some of our ideas with thoughts and what is going on regarding research and on SCA10, on Spinocerebellar Ataxia type 10. I'm also glad that this session or that this month has two sessions. The second session will be conducted by my friend and colleague Birgit Schul. So we agree to not combine and not to overlap on the topics. So I will, in addition to the topics that I am assigned for, I will also include a little bit about the clinics and the epidemiology of SCA10 as well, and the advanced genetics components, some of the advanced genetic components, especially regarding the structure of the repeat expansion, will be largely covered by my colleague Birgit Schul next Monday. So you're very welcome also to join that session. So the idea for the agenda for today is I divided all the session into 6 topics, starting with an overview of what Spinocerebellar Ataxia type 10 means. We will discuss a little bit about epidemiology, considering that so interesting and unique key things that we have on epidemiology of SCA10, and we'll also cover a little bit about briefly regarding the main clinical features of SCA10, and we will move on obviously to the current state of the research and clinical trials that unfortunately, and I can say that in advance, there's very limited information, and we need to, move forward on this aspect in SCA10 compared to other SCAs that were discovered by the time of SCA10. Probably in SCA10 we are not so advanced in terms of clinical trials or therapeutic approaches, and there are many aspects that needs to be solved before to move forward on that aspect. And finally, I will try to share with you some some principles of managing SCA10 considering our current situation. So let's start. So a Spinocerebellar Ataxia type 10 or SCA10 also, and there's another initiative of nomenclature for some of movement disorders with genetic cause that calls like SCA10 as SCA, referring to the main phenotypic feature, meaning the ataxia, together with the name of the gene. So that's another MDS initiative from the International Society of Parkinson's Disease and Other Movement Disorders. So SCA10 is an autosomal dominant spinocerebellar ataxia, and it's a repeat expansion disorder. In case of SCA10, the repeat expansion is a pentanucleotide. It is an ATTCT repeat that it's located within the intron 9 of the ATXN10 gene located in the chromosome 22. It is important for you to know, and I would like to show you, despite this busy slide, just for us to remind that there are, a lot of disorders that are repeat expansion disorders, and all of them have the genetic causes in different sections, in different regions of the gene. Some of them are located in coding regions related to the formation of specific proteins, especially the polyQ disorders, including many SCAs, for example, and Huntington's disease. But as you can see here, in the case of SCA10, we are talking about an expansion that is actually located in a non-coding region. And that's also very, very important to keep in mind, also for the clinical aspects and also for the therapeutic approaches that we have to keep in mind. It's also important to remind that SCA10 was probably like the very first pentanucleotide repeat expansion that was discovered from now about like 25 years ago. And at the same time, because of probably this disorder is mostly reported in Latin American regions, and a couple of case series on East Asian regions from China and Japan, and one report from a Sioux Native American from Minnesota in the US, it means like there's only very few regions across, in the world where SCA10 is actually meaningful health problem. So probably in the other regions of the world, SCA10 is still considered in the section of very rare disorders or even not non-reported disorders. So what about the biology of SCA10? So SCA10, as I just mentioned, is a pentanucleotide expansion, and this expansion is located in an intronic region. It's an intron 9 of the SCA10 or the Ataxin-10 gene. And as you can see here, it's important to keep in mind some things, because there are other repetitive fragments that are also part of this intronic region, and it includes this line 1 repetitive fragment that in the evolution of this gene, so it's suggested that this line 1 repetitive fragment was first inserted, and after that, right in the middle of this line 1 repetitive fragment, an ALU fragment was also included, and as part of the tail of this ALU fragment, we are supposed to have this repetitive expansion of SCA10. Here you can see this SCA10. And that's also important because many of the intronic repeat expansion disorders have this relationship to these ALU components in the intronic regions that might have not only importance in terms of understanding the evolution or the origin of the mutation, but also might have some importance for looking for therapeutic approaches after that. So because of that, this pentanucleotide expansion originally, if we talk about evolution of this fragment of this motif, so it comes like over time across like generations, we probably have this repetitive fragment, this motif, this ATTCT, from 3 different types of mutations. Can be direct mutations from the ancestral motif that it's to be suggested to be a TTT 5 times T motif, or other successive mutations, changing one nucleotide first and changing the other nucleotide as a second mutation to get this common ATTCT fragment. Or also it's a conversion from the ancestral ATT-TT variant. So normal population not related with SCA10, usually if we measure we size this section of this expansion in the normal population, usually they have between 9 to 32 repeats. That's what we know until now. And patients with SCA10, ataxic patients with SCA10 usually have from 800 to 4,500 repeats. So it means it's an expansion that it's really huge. And it's really important to keep that in mind. There's also a low penetrant isolated case that was reported with 280 repeats, but most of them are over 800 repeats. That it's also important to understand because it also clearly shows you that there are some lack of information regarding other kind of alleles. What about alleles between 33 and 279 repeats, or even alleles between 281 and 799 repeats? So until a few years ago, there were no information and no reports regarding people, from the normal population harboring this kind of repeats that we now consider a kind of intermediate alleles. And so this is a recent study that we conducted in the Peruvian population, where you can see that over 800 participants from the normal population that we were able to recruit and genotype for this expansion, we found about 9% of all this healthy Peruvian population harboring more than 32 repeats. And that's even more interesting that if we split this Peruvian population into, on the one hand, mestizo population that were recruited mostly in Lima, the capital city, and Amerindian population, meaning people from certain regions of our country where we really know that the percentage of Amerindian ancestry is above 90%. So apparently the percentage of these intermediate alleles, it's even higher in the Amerindian subgroup of the Peruvian population. What's the meaning of these intermediate alleles until now? Really, we are in the path of trying to understand what does these people harboring these intermediate alleles means. But at this point, you can see that it's really important to check that if you, for example, are testing a patient with suspicious of SCA10, and this patient has a high Amerindian ancestry, or by history or by ethnicity information, has a kind of combination with Amerindian ancestry, keep that in mind, because it's gonna be really hard to clearly diagnose SCA10 just based on some non-quantitative techniques for genotyping that we will discuss right after that. And so regarding the pathomechanism in SCA10, we have to keep that in mind that still until now, we are not clear about what is the underlying pathophysiology on SCA10, unfortunately, but there has been many hypotheses that has been proposed, and especially I highlight this one that was proposed by Professor Achisawa lab, that recognized that as any other intronic repeat expansion disorders, probably in SCA10, the main physiopathological component is regarding an RNA gain of function. So meaning that the problem is not the product, the protein product of the ataxin gene, But the problem are the gain of function of the transcripts of the RNA of this ATXN10 expansion, because these RNA accumulate and form some groups in foci that are located both nuclear and also in the cytoplasm of the cells. And this kind of RNA foci capture or trap some ribonucleoproteins, including or especially the ribonucleoprotein K, that over time leads to an activation of apoptosis pathways that finally ends on a neurodegeneration process, including the component of epilepsy that it's also described in SCA10 patients. We also have to keep in mind that this model, that this hypothesis needs further information. There's other kind of studies that needs to be conducted both in patient-derived cells and also in other animal models that probably confirm or adjust this preliminary hypothesis that it's also important. Or in addition, based on both information that we have on SCA10 patients and also based on mouse models for SCA10, we know that these transcripts, that this mRNA is also expressed both not only in the human brain, but also in other tissues, including the skeletal muscle, the kidney, and also the liver. No? So, regarding the epidemiology of SCA10, so it's important to give this kind of overall world frequency of the main SCAs that it was published in 2019. There's over the last 5 years, there are more repeat expansion disorders that were recently discovered. But the idea of this map is for you to highlight where are overall the SCA10 cases, and as is clearly for you to know that SCA10 is reported mostly in the American region. There are also cases reported in China and Japan, as I previously mentioned. But what is really important here at the world level is to understand that the large cohorts of other SCAs from Europe or even in North America or in other regions across the world failed or didn't identify neither one case of SCA10. So really SCA10 is a kind of really restricted disorder to certain regions across the world. And if we focus these regions in the Latin American region, so you can see in this map that SCA10 was mainly reported in the Latin American regions. The very first two cohorts described of SCA10. The very first one was described in Mexico back in 2001, and followed by many of these reports conducted in Brazil, especially in the region of Paraná, in the group of Professor Elio Teibe, and also in other regions in Brazil, in the north and also in the south. So these two were kind of the main cohorts for quite a long time for SCA10. Just recently, our group published a large cohort of SCA10 patients in Peru as well. The others are mostly case reports of isolated cases that we have with SCA10 described in many other regions across Latin America. And this other map that you have here in the right side of the slide, it's for you to know that how frequent is SCA10 in some of these countries. So SCA10, for example, in Mexico, it's considered among the identified dominant ataxias, the second most commonly diagnosed or identified SCA after SCA2. And in Brazil, and that has to be, you need to to understand Brazil is a huge country, but overall we can say that probably SCA2 is kind of the second in some regions or third position in terms of frequency among dominant SCAs in the Brazilian country. But in Peru, and based on the cohort that we recently published, is SCA10, it's largely the very first and the most common identified SCA in our population. And that is kind of interesting to know, because we really, if we wanna move forward on looking or understanding the mechanisms of this disease, and after all, looking for therapeutical approaches, probably one of the things that we need to do is try to understand and to characterize better other cohorts in addition to the Brazilian and the Mexican cohort. There is, based on that observation and that initial observation, that it has been hypothesized that SCA10 is actually an SCA that is considered of having a Native Amerindian origin, and mostly based on the two following findings. Mostly reported at SCA10 cases are from the Americas, including this Sioux Native Amerindian, and also from the East Asian, I was telling you, from China and Japan. It was actually not found in large European cohorts. And so considering especially this Sioux Native Amerindian means that probably the origin of SCA10 goes back to the population of the Native Amerindian population that was even before the Spanish or the Portuguese conquer. So that is why we track the origin of SCA10 on this period, 15,000 to 20,000 years ago. And because of the migration patterns that we have by that time, that's why we have this spread of cases across the Americas. If we talk about clinical aspects of SCA10, we have to keep in mind that the most or the vast majority of information that we have until now for this are based on the Mexican cohort and the Brazilian cohort. These are in the case of the Mexican cohort, this is the original paper published in 2001. They published 4 families with very detailed clinical information. And one of the things that gains attention in this first initial report is the presence of, atrophy in the cerebellum, as you can see in this MRI image, but also a high frequency of seizures in these Mexican families. And overall, after that, there were subsequent publications that consider up to 72% of the Mexican SCA10 patients actually reporting some time of seizures. On the other hand, the Brazilian cohort, since the very beginning, and this is not the first case, but this is the largest case that consider 91 SCA10 patients from 16 families, is still have what they proposed since the very beginning. A very low frequency of epilepsy and seizures, goes up to 6% only in the Brazilian cohort. So that's why there was a huge contrast between the Mexican cohort with large and very high percentage of seizures, contrasting with this Brazilian cohort that only shows very low frequency of seizures. And if we can add to this information the information that we have until now on the Peruvian cohort, we can say that by now we have 116 patients in our registry of SCA10, all of them coming from 75 non-related families. And as you can see, this is a map of our country. And most of the patients are located, obviously, in Lima, the capital city. For those not familiar with Peru, the capital city of Peru, Lima, concentrates 1/3 of the Peruvian population, so makes sense that have many cases from there. But what is interesting, and we noticed that since our first report of the very first 3 families, it was in 2013. Most of these families were located in the highlands region, in the central and southern part of our country, and that was also overlapping with the ethno-linguistic region that is more influenced by the Quechua language, that is our native language, suggesting again this Amerindian origin. So regarding the clinical pictures, you can see there are some cases that I show here. Our patients kindly allow us to show that for academic purposes, but you can see that most of the cases obviously have ataxia. They have gait instability and all the cerebellar signs. There's a kind of low progression, but there's also other abnormalities, including ocular abnormalities, some of them with neuropathies or neuropathy disturbance as well. And we didn't show that, but there were a frequency of about 14% of seizures. That is kind of in more than, for sure, than the Brazilians, but less than the Mexicans cohort. Let's go. So other clinical aspects in SCA10 includes, like, for example, the ataxia usually starts at age of the earliest case reported at age 14, but usually there are late onset, adult onset disorders until 45. It's kind of a very slow progressive ataxia overall. There's a published that we have comparing the disease progression of SCA10 with SCA3, for example, together with the Peruvian and the Brazilian cohort. And clearly the progression of SCA10 is much less that is more slowly progressive if we compare at least with SCA3. There's also evidence based on many pedigrees that suggest incomplete penetrance in this disorder. And in addition to the motor symptoms, that most of them are related to the cerebellar signs, it's important to highlight these seizures. And probably, Birgit, we will discuss more about that, because one of the proposed hypotheses to explain these differences in the frequency of seizures between Brazilian cohort and the Mexican cohort, for example, suggests that the purity of the pentanucleotide expansion and the presence of these interruptions, these other motives different to the canonical ATTCT, may be involved on the presence of seizures. And so having these other motives within the tract, ATTTC, or these other mixed interruptions, might be increasing your risk of having seizures if you have this expansion. In addition to these seizures, there are evidence that also reports cognitive impairment, especially, non-cortical cognitive impairment pattern in SCA10, and also neuropsychiatric symptoms that are kind of largely variable. There's a recent paper that just was showing a very disrupting case with a kind of schizophrenia phenotype, for example, in a patient harboring a very large SCA10 expansion also. And so those are the most clinical features. And in terms of other studies that has been conducted from the clinical perspective, it includes like MRI volumetry, for example, and based on that, we can say that the atrophy that it's confirmed in SCA10, and this is information mostly from Mexicans and from Brazilians, are cerebellar atrophy, including a little bit of mild cortical atrophy, but as you can see here, it's mostly located at the cerebellum, the atrophy, and also in some other nuclear ganglia, including the pallidum, for example, that are also with some evidence. There are not too many post-mortem SCA10 brains. As far as I have knowledge, there are only 2, and this is one of them. And so based on the information on neuropathology assessments performed in post-mortem SCA10 brains, we have this symmetrical cerebellar atrophy, and there is a clear predominance of Purkin's cells that are the mostly affected in SCA10, and also the molecular layer of the cerebellum, with no big changes, at least from the neuropathology perspective, in the cortex or in the hippocampus, midbrain, or pons. And so kind of in correlation what we was also suggested with neuroimaging. So as you can understand, in SCA10, since we have this very huge and very large expansion, so diagnosing SCA10, it has been not an easy task. And there are many techniques for genotyping or sequencing, methods for genotyping and sequencing this repeat expansion. All of them are here. But what are highlighted in red are those methods that at some point are used, for example, for SCA10 diagnosis, including this repeat prime PCR method that we have to understand that it only has a qualitative approach, meaning it's not possible based on this technique to know the exact number of repeats of this pentanucleotide expansion. We can say the number for normal alleles up to 50 probably, but no more than that. And the problem, so the gold standard for quite a long time for SCA10 was the Southern blotting, but you all know, or many of you might know, that Southern blotting is a very complex method and sometimes a little bit expensive, and also includes some regions that are kind of risky. And so it's kind of not a standard now for that. So the new long read sequencing technologies, both like nanopore, for example, or PacBio technologies, both of them are starting to help to sequence these large expansions like SCA10, and hopefully we can use this kind of technologies together with others like optical genome mapping that might help in the future for SCA10 diagnosis. At this point, these 2 technologies are available, yes, starting, but not, for example, being used for diagnosis for patients, are only research-based. And let's move forward on how do researchers study this disease. And so some of the information that I already discussed with you were information that were obtained, for example, with animal models. And in SCA10, we have at least 3 types of animal models. For example, we have this knockout mouse, meaning that we blocked in this mouse the SCA10 gene, and we confirmed that, for example, these mice with knockout with no SCA10 gene goes to embryonic lethality. And so that is important to know. But in addition, if we have this heterozygous ataxin 10 mouse model, for example, there are no motor phenotype in this model, meaning that decreasing in 50% the dosage of SCA10 proteins actually doesn't change the phenotype, at least for the mouse model. And we also have this transgenic mouse. For example, this one has included a 580 TCT expansion. And with this kind of mouse models, we were able, for example, to confirm that there are these RNA foci, and also to confirm a neurodegeneration pattern that, however, we couldn't confirm with this model a cerebellar pattern. And so that is also still a problem with SCA10 in terms of models. Probably other animal models are required or has to be refined in any of these animal models to obtain a better mimic of what's going on in humans. And in addition, we are also working on somatic cell lines. And based on somatic cell lines, we have the information that, for example, their large expansion do not impede transcription, and that makes sense. The mutation, the expansion is not in a coding region. But it was also important to check and to verify and to confirm that. And also based on these somatic cell lines, we confirm this presence of RNA foci, both in nuclear and also cytoplasmatic foci. There's also initiatives going on for stem cells-based modeling for SCAs, including that in SCA10 as well. And so those are the kind of models that we are at this point using or taking advantage of for SCA10 research. And in addition, if we as patients, and that's, I think, important for patients and families, if they want to be involved in research initiatives. So what is important for contribution and to understand, there are still many things with needs to better understanding in SCA10. So participants are required to donate, for example, biofluids, including plasma and CSF. From the spine. We can also obtain cells, from the fibroblasts, from lymphoblasts, from myoblasts, are all of them, and also stem cells that can be used for, generating other cell lines, including neurons. And I left the first thing that is called tissues for the last thing. So it's really important perhaps to try to understand, and that's for all the SCA10 community, that we really need to understand better the pathology of SCA10, and that can be done only, for example, with tissues, brain tissues, for example, post-mortem brain tissues. So that's, for example, in Latin America, at least, it's a very sensitive topic for patients and for families, and needs to be discussed with the families, so they probably in near future can understand the importance of this kind of research based on brain tissues. On the other hand, the national history or natural history studies are also required for SCA10. We have information, as I just mentioned, only based on the Mexican and the Brazilian cohorts mostly. We are starting with the Peruvian cohort at this moment, and the clinical natural history studies are longitudinal designs that also consider or include a lot of biomarkers, including clinical biomarkers like the SARA and NESCA that are motor and also mixed motor and not motor scale assessments. And we also need assessments for cognition and neuropsychiatry. And I think we make huge progress, for example, including the huge efforts of National Ataxia Foundation, trying to promote and to train both scientists and also clinicians on applying correctly these kind of scales on clinical settings and also for research purposes. Biomarkers are also important, and in addition to the biofluids that I just mentioned before, we also need biomarkers including MRI. I only mentioned information regarding volumetry, but recently, and that's very interesting information regarding other SCAs, for example, spectroscopy, protocol of MRI are also required to understand and will give us some information that might be made helpful, this kind of biomarkers, if we wanna use them for clinical trials, for example. And so that goes close. We're also getting to the end. And unfortunately, in terms of therapies in development in SCA10, we are not advanced as other SCAs, for example, like Machado Joseph SCA3, that previous speaker in webinars from NAF just showed like at least 25 different therapies in development. This is information that I captured from the NAF, from the National Ataxia Foundation website. And for SCA10, we only have information on these two molecules. Actually, the first one was not specifically designed for SCA10. It's considered here because in the clinical trial, they include some SCA10 patients with other SCA10. So this troriluzole, as you know, it's a pro-molecule of riluzole, and it's related to this glutamate regulation that can prevent exotoxicity in the cells. And so they are actually on phase 3. And there is another molecule that is, still in development that you can see here that focus, and that makes, based on what we just recently discussed, trying to address this RNA component based on what we until now know regarding the pathophysiology of SCA10. And in terms of therapies, there's no more initiatives until now that I am aware of. Just want to end with some comments, and this is probably most for the families and for patients living with SCA10, because given that there's no specific curative or modifying treatment for SCA10 until now, it is important to understand that patients are now, not tomorrow, not in the future. And so when clinicians deal with patients living with SCA10 and their families, probably they have to consider these principles. So that the management has to be obviously a multidisciplinary approach, but in addition, an approach that has to be oriented based on the patient needs, but also on the family needs, on the caregiver needs, that not all the time are aligned. So the needs from these three groups not always are the same. And so it makes sense for clinicians to take the time to request information and to ask about one of each group specifically to try to help families living with SCA10. The therapy right now is just basically symptomatic, and that include the use of anticonvulsivants, as you know, because of the seizures. But over time, it's important to discuss with patients if some of the medication that we use as symptomatic are really helpful, especially when we are in advanced phases of the disease. So probably this reduce or even discontinue some medications can be even more helpful depending on the stage or of each case, obviously. In all of cases, physical therapy and nutritional support are important, and they have to be, the basis of the treatment, and obviously the genetic counseling, not only for the affected participant, but also for all at-risk family members. And with that, I want to thank you, and I'm really open for questions and to listen your comments and thoughts.
strength in the lower body for folks who have SCA10?
Yes, for sure, it's a good idea, and I think this kind of training and it's not only applicable for SCA10, probably applies for every type of ataxic disorder in not only the genetic form. So really, I think it's a good thing, and there's initiatives that we probably can consider in the future, not only based on genetic therapeutic approaches, but also rehab medicine approaches that can help patients on daily living on specific difficulties they have probably needs to be tested under, a scientific methodology design
Yes.
that needs to be done. And not always it's an easy to do these kind of designs, but probably we need to work on that.
Thank you. Our next question is from a participant who asks, are intermediate repeats independently possibly pathogenic or only if they expand? Can you explain how 280 is pathogenic, but not those with similar repeats?
Yeah, it's really, well, the short answer is we don't know yet. So that's the short answer, but I wanna expand a little bit. So based on what we know before, we published the information, for example, of the Peruvian cohort. There was no information regarding these intermediate alleles in the normal population. So we just found these intermediate alleles in normal population. So what these alleles means for these participants, that's a real question, because there are many possibilities. One of them is that these intermediate alleles are just low penetrance or even not really significant in terms of develop any type of sign related to SCA10, but also might be possible to have a sort of mild phenotype on a very late stage or a very late age of these patients. So we need to follow, for example, these patients over time. And the other problem with these positive intermediate alleles is like, if you use only, for example, an RT-PCR methodology to diagnose SCA10, and you say this is above 50 repeats, and this patient or these participants actually only have an intermediate allele, you can misdiagnose this patient with SCA10 when probably it's just an intermediate allele. That's the huge problem, and that's why we really need to sequence and to really be accurate about the size of the expansion. Hope that helps to answer that. And we are in that, so really we are trying to study that now.
Oh, it's good that to know that work's being done to help answer some of these questions. It just research takes time.
Yeah.
Our next question from an attendee who asks, how can I find out if my wife's SCA10 diagnosis was reported? So participating in maybe some of the research that you were talking about. She is from California and was diagnosed around 2006. Her older brother was also diagnosed about 5 to 10 years ago, and they're of Guadalupe descent. So is there any way for them to know if they've been involved in this kind of research or to check?
Yes, well, that's another ongoing process work because there are initiatives I didn't have the time to show, because in addition of the clinical trials, we have about like 10 observational studies that are currently ongoing for SCA10. For example, there's one in Brazil, at least one in Brazil that I am aware of. So people that lives in this region can contact, for example, some of the lead researchers, and for example, National Ataxia Foundation, and I'm glad to do that, publish and have information on the website regarding these opportunities. Some of the researchers, for example, we usually publish on advocacy groups or family websites regarding specific ataxias, short information inviting people to participate on specific studies, including the selection criteria for this. So we are also in Peru, for example, inviting all SCA10 participants and families to contact us, because we are trying to expand our SCA10 cohort and trying to, start with a natural history study, a longitudinal study, and to understand better how's the progression of this disorder. For people living in North America, so there are centers that are working in SCA10 for sure, and that can be contact. For example, Professor Tia Shizawa at Houston Methodist, and Houston also have extensive work on SCA10. He or his colleagues working with him are also probably interested. And other NAV centers of excellence, I'm pretty sure that NAV centers of excellence are also a good contact that you can reach and try to ask them if they are familiar or they are aware of any particular research on SCA10.
I'm also going to put a link in the chat that's a link to Dr. Ashizawa's study to learn more about it if you are in North America and would like to connect about research.
Exactly.
And our next question is from someone who asks, what are the cerebellar neurons damaged in SCA10? Are granule cells of the cerebellum also involved?
Well, what we know until now is like the most affected cells in the cerebellum are the Purkinje cells, and mostly there are kind of direct damage on these specific cells, also cells from the molecular layer that I mentioned in my slide. But we really need more information and more studies to really understand with more participants, with more animal models, for example, to check what is going on specifically on SCA10. But apparently there's kind of very focused damage and not as a spread damage across the cerebellum.
Very good to know. Thanks for explaining that. Our next question is from someone who asks, are the seizures you mentioned, are the seizures focal or generalized, and how difficult are they to treat?
Oh, that's a good and very interesting question, a very good question, because for clinician that is very important. And well, the seizures are different, so there are reported both. There are generalized tonic-clonic seizures, but also focal that seizures, and also focal with secondary generalization. But the good thing about SCA10, at least on the vast majority of patients with seizures, they are easily treatable. So you use one or anticonvulsive medication. You don't need probably in the majority of them to combine medications. So they are not, hard to treat seizure syndromes. But it's also important for you to know, and I forgot to mention that when you have a patient with SCA10 with seizures, or even a patient with SCA10, please take a look at the rest of the family, because there are reports, and we found in our families that there are other family members not having ataxia, but having seizures without ataxia. And so we are still we need to understand if there's the possibility of, having only a seizure type phenotype on these SCA10 families.
That's very good. Thanks for explaining that. And we have another question. Could you please comment on the lifespan of people who have SCA10 and whether or not repeat expansion to sizes is related to age of onset or severity of symptoms?
That's another excellent question. So regarding lifespan, so as I mentioned, the start of the disease goes from 14 to 45 years of age. We know, at least from the very few studies that we are involved and we recruited, that there's a very slow progression overall on SCA10 patients. So we do not expect, a very hard limit of life expectancy of patients, but overall, that is the problem, that they are pretty much disabled, and over time, the causes of death are mostly related because of that, because of the disability, pneumonia, aspiration, and so on, that we also know in the majority of patients with advanced ataxias. And there's no information about longitudinal studies that I am aware of that are following, for example, patients for decades that can accurately answer your question. But yeah, we need that also. And regarding what was the other question? There was a second part of...
It was whether expansion size is related to age of onset or severity of symptoms.
Yes. Well, there's a huge problem because of that, because there are some studies that try to correlate the age advances with the size of the expansion. And so in some of the participants with SCA10, we found an inverse correlation that is mostly described in other type of SCAs, but in others don't. So that is the problem. So in some of them, yes, in some of them don't. And in addition, you have an extra problem that sizing the repeat expansion, as I mentioned, it's really hard, and you have to really be careful about and accurate about this. So until now, it's probably an evolving knowledge that we need to address with these new technologies of long-read sequencing and redo all of these correlations with these technologies.
And we have time for one final question. We have an attendee who asks, are there any vitamins or supplements that you think would be helpful? And this attendee is sharing that he takes vitamin D supplements and he finds them beneficial.
Well, that is also important for clinicians and so on. So I have to start to say that there's no evidence that any other supplement or vitamin or whatever, it's really helpful. I mean, with evidence in SCA10, unfortunately, but based on what we do with other SCAs and what the limited evidence we have. So I think in general, it's important to have very good lifestyles, for example, so have good food in terms of variety fruits, salads, and so on. It's also important because increase your level of vitamins or antioxidants. That there's some evidence that says that helps a little bit. So there are other supplements, like for example, what is the name of this? It's on the top of my hand and my tongue, and I don't remember the name. I will come back with that. But these kind of supplements are a kind of antioxidants that you can use for patients. For example, we use in some patients, but there's no strong evidence of they to be helpful, but there's a strong evidence that they are secure. So they don't put in risk the patient. So at least if there is a little possibility to be helpful and they are secure and safe enough, we sometimes decide to use them. For there's others, and more than once I received information from patients with a lot of products, some of them very fancy products. And so what I always answer the patients and families is, and I suggest that to clinicians dealing with patients with SCA, try to not be strict with your answer, because always families and patients, we will look for all things that they think that can be helpful. The family members always give suggestions and so on. So I decided just to say, so I will check. So give me the information, I will check the evidence for this product, and I will double check about the safety of this product. If the safety of this product is kind of strong and you still want to take that on your risk, so you can do it. But probably I just want to make sure this molecule or this product won't put you in risk.
That makes a lot of sense. And we are out of time. So thank you so much, Dr. Cornejo-Olivas, for spending this last hour with us and telling us more about SCA10. We really appreciate your time and your expertise.
Thank you so much. And thank you for all the people that was engaged in this session. Thank you so much. Bye-bye.
Yes. Thanks to you for attending this session. I hope you've learned something valuable today, and wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, everyone.